Sure. So this is a collaboration that we started with a company called Repair, and PKMYT1, it was one of the targets that they identified that could be examined in tumors that have high levels of cyclin E. So over the years, we have developed several model systems for which cyclin E is overexpressed, and these model systems span both cell lines, transgenic mouse model system, as well as PDXs from breast cancer patients in triple negative. So we were very interested in collaborating with them as they were with us. So we got some of the drug that they had developed, and then we examined PKMYT1 in our cell lines, and what we found is that in fact when cyclin E, and in our case the form of cyclin E that was is tumorigenic is the low molecular weight forms of cyclin E, which is generated by the elastase, neutrophil elastase cleavage sites at the N-terminus. So these forms become oncogenic, and these forms have the ability to make the triple-negative breast cancer a lot more metastatic or aggressive. And what we found is that these cells respond, they're very sensitive to inhibition by the MYT1 inhibitor. Their MYT1 inhibitor is called RP6306. So we were curious to see what happens also in our breast cancer patients. So we had done a clinical trial with a neoadjuvant chemotherapy in breast cancer, triple-negative breast cancer patients, and we had samples from those patients, and we had stratified them according to cyclin E low and high. So then we then ask, so if cyclin E levels are high, do we also have high levels of phospho-CDK1, which is the target of PKMYT1? And in fact, the correlation was very significant. So it was suggestive that triple-negative breast cancer patients who have high levels of cyclin E, especially the low forms, and have high levels of phosphorylated CDK1 would be good patient cohort to respond to the agent. So we went on and examined mechanism of inhibition through DNA damage and premature entry into mitosis at the cell line level. We looked at about 7 different triple-negative breast cancer, and we also knocked down cyclin E, and we found that under those conditions, the cells no longer responded to PKMYT1. From there, we moved to our transgenic mouse model system that we had developed in our laboratory that have the low forms of cyclin E, and we can see that, in fact, under those conditions, they do respond very well to PKMYT1. And then we also examined our triple-negative PDX models that we had parsed according to either high or low cyclin E. We had models of both, and we found that in those cases that had low cyclin E, they didn't respond at all to PKMYT1 inhibitor, but the ones that had high levels of cyclin E, they did. So collectively, the results suggest that in cells, tumors, patients that have high levels of the low forms of cyclin E would be good cohorts to respond to RP6306, which is the first-in-class PKMYT1 inhibitor from Repair.